Hi, everyone. Welcome to the fourth seminar speaker for the Warnell Seminar Series. And today, for our speaker, I'll have one of my PhD students, Hannah Petronic, introduce him. And I just want to put a plug here. I'll let her introduce him because she's really the person who, bugged me and say, hey, we should invite this person and then organize everything. And so I want to give her kudos for that. But also, as a reminder, if you all want to have, a speaker here that really is someone important in your profession, you could look like Hannah. Go plug your PI. We cannot accept nomination directly to graduate students, but we can have, a graduate student back in their PI and their PI coming to me. So again, it could be you next time presenting someone who's very relevant to your field, because that's, the number one complaint I hear about the seminar every time. There was not a speaker that talks of my stuff. I'm like, well, did you invite them? No, so I cannot invite them. So please, we're always looking for nominations. Go back your PIs if they're not already nominating people and, propose them names. Have them identify the name for them of someone that you think will be very nice to have here. That if you think is a great researcher, great speaker in your discipline, and, your PI will be happy to come to me, and usually we have room. So that's, again, just a little plug for future nominations. And Hannah, I'll leave it to you.
All right. Hi, everyone. I'm going to go ahead and introduce our Warnell Seminar speaker this week, Dr. Brian Lovett, who's visiting us from the state of New York, where he's currently serving as the Chief Data Officer for the nonprofit Stand Up for Science, and is also an adjunct faculty member for the entomology department at Cornell University. Dr. Brian Lovett earned his PhD at the University of Maryland and then moved to work as a postdoc in the at West Virginia University, where I had the privilege of working with him as an undergrad and for a little bit as a master's student. And some of the work he'll be discussing today earned him the Little Prize in 2019, which is for the most outstanding publication in the Journal of Science. Well,
thank you very much. Thank you. I am so glad to be here to share what is essentially just a summary of a bunch of really cool work that I have been able to do during my career. You can see from the title, it's going to be a little bit unhinged, but I'll walk us through everything. Before I jump into the science, I do want to take this opportunity and this platform to make a broader point, which is that a year ago, I was a USDA researcher in Ithaca, New York, and I led a lab of some wonderful, talented scientists. And I was one of the scientists that was targeted by the government to have my lab closed. And so I just want to provide a cautionary tale on this. This, in February 13th, is the email that they sent me that in one line said, you're fired based on performance. I had never received any negative performance reviews, but they were using this as an excuse to get rid of the new PIs. And so I had worked at the USDA for 2 years and about 9 months. And my job doesn't become secure as a USDA research scientist until year 3. And so because I was not essentially 3 months later into my career, they were able to fire me in this fashion. Later, courts determined that it was illegal. And so they sent me 10 days later an email saying, great news, you can work for the agency that just fired you. And so I rejoined them, but ultimately had to make a really difficult decision to move away from the agency. And this was not a decision that I made alone. They gave us a retirement, quote unquote, retirement program that we could choose to go into, and they were threatening to fire us. And so this is a group of 4 scientists, including myself, that comprised our USDA research group in Ithaca. Through my PhD, I had always admired this group of scientists because they led the way in insect pathology, and they house the largest collection of insect pathogenic fungi, live fungi that you could wake up and use to create biopesticides today. The 3 researchers here that I've highlighted in green, myself, Michelle Heck, and Lindsay Milbrath, we were research scientists. All we did was focus on research questions. Catherine Bushley, who I've highlighted in red, she was actually a curator, which is a different type of position, but not an active research scientist. 3 of us, Lindsay, Michelle, and myself, have all left the agency. Catherine Bushley is the only one who is left in that CRIS, and some of you may remember Catherine Bushley from interviewing for the mycologist position here. And so I just want to articulate that this type of broader effect and the thing that I underwent is something that many government scientists have gone through. It has changed the face of how our government agencies work and the type of science that can be done. And part of why I left is through the treatment that we had at that agency, I realized not only do my ethics not align with this organization anymore, but I cannot do the science that I want to do. And I think a great example of that was postdoc funding that I had secured for 2 years. I had selected the postdoc had accepted that offer, but because of hiring freezes, that money and ultimately that postdoc was pulled out of my laboratory. This is one example of stories across the nation, across the agencies. And as soon as I made this decision, I started to use my voice to speak out. And I spoke with Science Magazine about my experience and what happened to me. But the point that I made to them that they decided to pull out a quote, they even made this cute little sketch of me, although the beard, questionable. But government scientists, I believe, are canaries in the coal mine for American research. And the kind of treatment, the kind of changes that we underwent, these are coming and we're already seeing them come to funding agencies that we apply to and to our universities directly. And so just a cautionary tale there, something that I've gone through, and this is why, even though I'm still very fortunate to be affiliated with Cornell Entomology, I decided to move into the nonprofit space and work with Stand Up for Science. Happy to chat about that later, but I'm going to jump right into talking about the science. And so I am an entomologist, I'm an insect pathologist, and a mycologist. I'm interested in fungi that can infect and kill insects and how these different fields enmesh in the context of an infected insect. I love to find local solutions for insect biocontrol. I'm incredibly fascinated by fungus Arthropod Symbioses. I'm going to walk through so many different kinds of those during this talk. And just broadly drawing connections across fields, I find really interesting, and I always get really passionate about that type of work. And the reason why I think that drawing those broad connections is really interesting is because I kind of view our understanding of the natural history of insect hosts as a puzzle. Most insects, even very well-studied insects, there are gaps in our understanding of their biology. And those gaps, particularly in the cases of these entomopathogenic fungi, may be something that's really important for us to understand how that fungus infects that insect or how they encounter each other in nature. So I've done a lot of work looking into cicadas, and I want to walk you through how this host biology is related to the infection cycle of a cicada pathogen. So all of these cicadas that are in the hand of my colleague here, you can see they have these little yellow butts, this little plug on the back of the cicada body. We're going to dig into what this is. Mesospora cicadina is a highly specialized fungal pathogen of cicadas. Different Mesospora species will have different cicadas that they prefer to infect. So Mesospora cicadina broadly infects the periodical cicadas that emerge every 13 to 17 years in large broods across the US. But Mesospora cicadina is interesting, unique as a fungal pathogen because it doesn't just kill the host. What it does is it will infect the host, grow within its body, and then replace the back half of the host with fungal spores. But this cicada is still alive. And so while the cicada is alive and it wanders around the environment, it's spreading spores that can infect other cicadas. This type of manipulation, this development, is a really interesting kind of behavioral manipulation called active host transmission. But with Mesospora, this replacement, this really bizarre biology, I would argue, is related to the really bizarre anatomy of cicadas. And so one of the things about cicadas is they emerge according to particular environmental conditions. And so cicadas, as they develop through their biology, as they get close to adulthood, they will come up to the surface of the soil, and then they will sit in the surface of the soil and wait for that temperature to get to the right degree. And so these cicadas, naturally through their development, are sort of sitting in this topsoil space. This topsoil space is where they can encounter spores of Mesospora and become infected. And so where these Mesospora spores are deposited through the development of Mesospora is tied to this sort of timing of cicada development and emergence through the soil. So here you can see a cicada waiting for that thermometer to get to the right temperature. And all of these little holes here are cicadas that are sitting there and emerged out of the ground during the cicada pruning. Now, you're probably wondering what the heck is going on with this cow, right? There's an old joke in physics that if you want to understand a problem, first you have to imagine that your problem is a sphere. And if the problem is a sphere, then you can solve it with whatever easy physics equations you want. I think we tend to do this same type of oversimplification with insects, in that we tend to think about the inside of all insects as the same sort of sloshing hemolymph. Insects have an open circulatory system, so folks just tend to think of the blood of an insect being immediately available inside of an insect and sort of filling the body. But the reality is the insect's body is full of tissue, usually. In the case of cicadas, it's not full of tissue. And the most notable thing about a cicada, if you never interact with them, which hopefully everyone here has, is that they produce this amazing call to interact with each other. And so here's a video that I put into slow motion for you, where you can actually see these tymbals of the cicada snapping together and forming this call, which is actually very quiet. And you can hear in slow motion, it's not really that loud. But the way that it becomes loud is because the cicada's body is actually hollow. And it forms a resonating chamber. That small tapping of that organ can then be amplified. And so if we look at this development here, you can see essentially the entire abdomen of a cicada is full of air, particularly a male cicada, but female cicadas, it will be full of air as well. And so through this calling mechanism, the anatomy of a cicada is entirely different from most other insects. And so the environment that Mesospora has to navigate and ultimately express itself through is very different from other insects as well. And so when you are faced with a question like, how does an insect survive when its abdomen has been replaced by a fungus? Well, its abdomen is mostly empty. So the fungus is mostly taking up space that was empty in the first place. And so one of the things that has always been curious to mycologists about Mesospora is that unlike other members of this group of fungi, Mesospora produces spores inside the cicada. And this is why the cicada is able to produce that plug that I showed you in the earlier slide. However, that production is bizarre because most of these other fungi grow out of the host to seek oxygen or air and then produce spores in the air. But in the case of a cicada, you can grow into the cicada and find air. And so there's this natural shift in the behavior of the fungus relating to the anatomy of the insect host. And so this is why it's important to not think of every insect as just a sphere full of fluid. We looked at how this development works in Mesospora using a microtome sectioning of the insect host. This is actually a really great insect pathology technique that you can collaborate with veterinary departments with. They will do this type of staining for fairly cheap. And this allows us to monitor that broader development of the cicada. But you can see in this image how the plug of the fungus on the back half, the sort of left side of that cicada, is filling a space which is mostly empty in the middle of that host when you cut it open and you look at the cytology. And so this is something that was following up on work that was actually done in the 1920s, but we were able to show with very high resolution and modern staining and technology the development of this fungal pathogen in the cicada host. And so Mesospora forms two different types of spores. Canidia are the ones that form that plug that I showed you. But on the back half of their development, they produce what are called resting spores. Resting spores are an entirely different type of spore, which are durable and are meant to survive in the soil until the emergence of the next cicada. And so this is an example of where when the adults are up and in the air, the fungus is producing aerial spores that are transmitting through this sort of direct interaction from the plug. And when the cicadas disappear and go underground for, in some cases, 17 years, that fungus will wait in the soil for those cicadas to emerge again. Really bizarre, interesting sort of synchronization of the development of the fungus with the development of the host. The other thing that I have to point out here is up in the middle of this, I swear, simplified diagram of the Mesospora life cycle. There are cicadas that are mating. there's a really interesting manipulation that happens in cicadas that are infected with Zospora, where we see male cicadas that have an infection begin to behave like female cicadas. These cicadas will flick their wings and actually signal to male cicadas that they're interested in mating. And when those male cicadas try to mate with the plug of Canidia, they absolutely become infected with that fungus. And so we have a manipulation of the mating behavior by this fungus. We don't know exactly how that's being navigated from a molecular standpoint, but we know that that results in behavior that leads to increased transmission of that fungus. I just want to show what this wing flicking, that sort of click, how attractive that is to male cicadas. And so here I have a colleague who has a cicada on her hand, and she's just snapping her fingers. And you can see how the cicada behaves just hearing that snap. That's, whoa. And so you can see how, in order to implicate this, to understand what that means practically for like the manipulation of the cicada, we have to understand, and in cicadas we do, a lot about the behavior and the mating of cicadas to be able to say, oh, that's the mating behavior that the fungus is causing. In a lot of insects, we don't have that context, but we're lucky with cicadas that they're extremely parasitic. This type of manipulation is not a one-off from Zospora. There are entire groups of fungi that use the mating behavior of insects against them. And so this is an example of another fungus, Entomophora musci, related to Zospora cicadina, which infects flies. You can see which fly is infected with fungus because its abdomen has been distended with fungal spores. And in that case, the fungus actually will produce volatile compounds that are released out into the air that male flies will respond to and try to mate with. So this is a living male fly that is mating with a cadaver and certainly will become infected with Entomophora musci. Again, this is an example of fungus figuring out and leveraging the mating behavior of this insect host. And so looking at the manipulation of insect behavior has become sort of a bread and butter topic for my research program, especially working in the casting lab and on that cicada pathogen. I became very interested in how this works. And so we tend to break this down. And I articulated this particular Zospora cicadina style of manipulation in this pathogen article into three different categories. Fungi can have active host transmission, where the host is alive. Transmission is occurring while the host is alive. We can have summit disease, where the host is manipulated and is placed in a particular position, but then it's killed and the fungus transmits after it's killed. Or we have non-manipulative transmission. And I would argue there are probably a number of non-manipulative systems where they are manipulative. We just don't know enough about the system to articulate where it's manipulated. And so particularly active host transmission is a very like macabre and dark form of manipulation because they are taking this live organism and directing it to infect others of its kind. Extremely classic example of a summit. I have thought about particularly summit disease, this positioning of insect host in a elevated position. This process, we see a number of different pathogens across a diversity of not only insects, but arthropods can cause this kind of summited behavior, where the insect host, insect host is directed by the fungal pathogen, typically up onto a leaf, and then it is directed to bite down onto that leaf, so that when it's killed, its mandibles are actually holding its cadaver in place. And then this positioning, typically in an elevated spot, which is why we call it summit disease, is a good spot for spores to rain down and infect more insect hosts. And so they're actually moving that host into the ideal position for transmission of the fungus. How this works, why so many different pathogens are able to sort of stumble into this place, I have put together in this article a framework for trying to classify them. And so I break these summit diseases into either adult summit disease or juvenile summit disease. And in the case of adults, the behavior that I propose these different pathogens are zeroing in on is insect sleep. insects need to sleep just like us in order to survive and be happy. And so their positioning, how they hold themselves, how they protect themselves when they're sleeping, often recapitulates a lot of the stereotyped behaviors that we see in summit disease. Because when you're sleeping, you're in a vulnerable state. And so you typically want to hide yourself somewhere safe, move away from other predators. And in juvenile insects, I proposed a similar mechanism, which is leveraging the need of all insects and all arthropods to molt. And again, when you molt, you typically come out teneral, sort of soft, delicate. And so insects tend to go off somewhere and molt in privacy so that they can emerge as adults or that next instar without being disturbed. And so I think these are two examples, sort of natural behavior of insects, which could explain some of the things that we're seeing these different pathogens land into. There's a lot to be dug into here, but these sort of periods of quiescence are something that I think is important and is fundamental to all insects. When it comes to understanding how this is happening from a molecular side, comparative genomics has really been my major approach to break these things down. And so in the case of Mesospora cicadina, we worked with Jason Stajic's lab and his student Sharon Chu to sequence and start to interpret the genome of Mesospora cicadina. When we sequence the genome of Mesospora cicadina, we learned Mesospora cicadina has the largest sequenced fungal genome. And so the expansion of this genome is unique to fungi and is probably undergoing some sort of interesting evolutionary process that we can start to pick apart. Almost certainly it's related to the manipulation of the host and the high degree of host specificity that we see in these fungi, but we're just now starting to characterize what this is and dig into these problems. And so we were able to learn from this genome sequencing effort that across Entomopterales, this group of so-called insect destroyers, this massive genome expansion is a characteristic that they all share. And in fact, we know that they expanded because of particular transposable elements, specifically long-terminal repeats were amplified within this group. And so we're starting to characterize this and look at this more broadly, but we were able to conduct a much more comprehensive study just after that one, looking at the fly pathogen, Entomoptera musci. And with this, we were able to, again, show that the number of genes in these fungi are not massively expanding, but the size of the genome is massively expanding from those transposable elements copying and copying and copying throughout the genome. And we were also able to start to look at, in this case, I'm showing some circadian rhythm genes. These are probably important in manipulative behavior because we tend to see manipulation, for example, summiting, will occur at the same time every day. And so we started to look at this and say, okay, what genes are present in these fungi, what are contained, sort of keep track of this timing of infection. In my lab at the USDA, I worked with a fantastic postdoc, Kelsey Scott, to develop broader pipelines to accelerate our cataloging of entomopathogenic fungi genomes. And so this entailed development of a broader phylogenetic framework to actually look at the relationships of fungi, but also just developing tools in the Cornell and the USDA computing clusters to sequence and in a repeatable way assemble these different fungal genomes. And so with her working on the backside, I worked with a student up here on the right, Alex Lando, who is a student of Kathy Hodge at Cornell University, to massively scale up our understanding of the Entomopterales genomes. And so Alex would go and collect cadavers or sent cadavers of these Entomopterales. On this image here on the left, that is a single little insect here that has produced spores that are covering the lid. So those spores were launched out of that insect, covering that sort of entire lid surface. We can then isolate those spores onto a specialized media that we have to produce for these fungi. To create that media, we have to incorporate eggs and milk to have sort of animal proteins mixed into that media for them to be happy. And then from isolation, we can grow these up in Grace's insect media, which is a mimic of insect blood. And if we grow them up at a large amount of insect media, then we can move them through a broader sequencing pipeline that's generated to get not only high quality DNA from these hard to work with and hard to grow fungi, but get long stretches of DNA. And so we worked with Sarah Carpenter, who was a research scientist in my lab, to develop these molecular pathways to sequence these fungi. And we ultimately, for all of our into-mouth Thorales, push them into a PacBio sequencing strategy. PacBio, if you are not someone who's big into genomics, allows you to sequence really long stretches of DNA. So you have really big puzzle pieces that you can put together more easily. With our proliferation of transposable elements, if we did not use PacBio, it'd be very hard for us to put those puzzle pieces together, because lots of our puzzle pieces would be identical. And so this is sort of showcasing that collaboration, this broader goal. Let's generate these fungal genomes, and then bringing this team together to really make that happen. And so Alex has now sequenced 6 different fungal genomes in into-mouth Thorales. This is more than were available when she started. And we're able to start to look at the diversity within these interesting groups of, in this case, they're called aryneoid into-mouth Thorales. But these are all highly specialized pathogens, caddis flies, various flies, and ones that we were able to directly collect and sequence in life. Really exciting stuff. Really great to be able to put a molecular phylogeny informed by the entire genome onto this group, which has been very hard for us to pin down taxonomically. One of the things we were able to do with these broader genomes, which I think is really cool, stick with me here, is we took all of these different fungi that we were sequencing, and we ran them through this program called Busco. Busco basically tells you how complete your genome is. It is single copy orthologs, genes that should be present once in every genome. And you can use that to understand broadly how these fungi are related to each other. And you can say, if all 300 of these Busco are in my genome, then I did a good job sequencing that genome. But in the case of into-mouth Thorales, because they're such bizarre fungi, we kept getting very low Busco scores, which typically would mean you have a bad genome or your genome's not complete. But what we found is that if we sort all these different Busco genes according to which Busco gene it is, over on the right, the same Busco genes are missing from all of our into-mouth Thorales. So even though Busco is supposed to be used as a benchmark to help you work with your genome, we are actually starting to know enough about the into-mouth Thorales to say, actually, Busco is not going to work for us here. These are ones that we would never expect to find or never see from into-mouth Thorales. If you just have one genome, you can't really have that confidence, but with the number of genomes that we've been able to assemble here, we can actually start to understand the genomes at that level. So here, I'm going to transition to the vampire portion of my talk. This is work that I did during my PhD in the St. Ledger Lab at the University of Maryland. I studied a particularly interesting pathogen called Metarhizium. Metarhizium is a ascomycete fungus, which has a very stereotyped infection process. So here on the cuticle of the insect, Metarhizium will land as a spore. It will taste the outside of that insect and decide if it's an insect that it wants to kill. If it is an insect that it wants to kill, if it is in the host range, then that fungus will produce entire infection process to penetrate through the cuticle, using the compounds that it breaks down from the cuticle to further burrow into that insect until it makes it into the blood of the insect. Once it's in the blood of the insect, something really interesting happens. Instead of growing as a filament, it begins to grow as a yeast. The reason for this is because the insect immune system is going to have a really hard time finding a small cell compared to a long filament. And so as soon as it enters into the hemolymph and is in direct opposition with the insect's immune system, it changes its behavior and shape to avoid the insect immune system. And so what this means is that on the outside of the insect, the fungus is expressing very different genes from the inside of the insect. And so we can use this to take the promoters of those genes and use them as on-off switches. And so we can take the promoters of genes that are expressed in the hemolymph and use that to drive, in the case of our study, toxins inside of insect hemolymph. And so metarhizium in particular, and I've shown it in this diagram, it expresses this collagenous coat around the outside of it. Collagen is a very animal protein. And so the insect immune system will look at that collagen and be like, oh, nothing's wrong here. And it can continue to grow and proliferate. And so using this broader understanding of these fungi, we can then express toxins from arthropod predators to make our fungi even better at killing insects. And so naturally, metarhizium may take a couple of weeks, 10 days to kill an insect, but we can use these toxins to dramatically shorten the amount of time it will take for that fungus to kill an insect. And so for this research that I'm going to share with you now, I'm talking about a toxin from the Blue Mountains funnel-web spider here. We had a collaborator, Glenn King, who likes to poke these spiders and then take that little droplet and look at all the proteins that are in that droplet of venom. Venom is sort of a cocktail of different proteins, and those proteins will have different specificities. And so we can look at these and say, this is something that's going to hurt us. This is something that's going to hurt insects. In the case of hybrid toxin, it actually is broadly affecting neurons in insects. And so it is having this wide effect on insect neurons, and it's not just targeting one neuron. That's actually part of our design of this study, because chemical insecticides, which are typically used in mosquito systems, they tend to target the sodium channel. And what happens when you're targeting just one thing is you get a mutation in that channel, and then the chemistry doesn't work anymore. And so by using this sort of broad toxin from the spider, we're able to prevent that type of resistance in our technology. And so here we have a spider evolving for millions of years to figure out what the perfect protein structure is to kill insects. We can say thank you very much, and we can put that into our fungus. And so when we take a fungus, which is a mosquito pathogen, and we drive a insect-specific toxin under a promoter that only turns on inside of insects, we find that mosquitoes are killed faster when that toxin is present, and that mosquitoes are killed with a lower spore dose. What this means, killing in a lower spore dose, practically, is that in the field, even if you have very few spores surviving, you could still kill that insect. And so over time, that low spore dose means that treatment would have what we would call increased persistence, which means it will last longer. So it's really great when something works in the lab. Very exciting. You can sometimes publish that even. But that does not mean it's going to work in the field. And so we developed a collaboration with Abdoulaye Diabate in Burkina Faso in sub-Saharan Africa. And we worked with him to develop a facility to see if we could have these same results that we found in the lab under field conditions. Specifically, we built a facility which we called a mosquito sphere. Here are some of our baby pictures of the sphere being developed. So you can see inside each compartment, we had a house. We had vegetation surrounding that house. We had a little moat to prevent predatory insects from walking in and around that house area. And we had 4 compartments that were experimental. And then we had 1 compartment, which was for rearing insects, mosquitoes that we captured, and 1 compartment that was a general space for keeping cattle. When you're working with mosquitoes, you have to have a blood source. And in Burkina Faso, you can use cattle to attract the mosquitoes. And so that was a way that we could have blood feeding in our experiments without having to have someone volunteer to do that. And so this facility was really amazing, allowed us to conduct a number of experiments that I'm going to walk us through. But the major objective that we had is to monitor, does our transgenic fungus have higher efficacy compared to the unmodified fungus under these semi-field conditions? And so the way that you can think of this sort of semi-field setup is that this facility is like a very large greenhouse, but instead of having glass, it has a double wall of mosquito net. So that allows environmental conditions somewhat to come into the sphere, but prevents anything that we're releasing or working with from leaving that facility. This is a fairly standard approach for broader entomological techniques, but this was first of its kind movement of fungal biotechnology. And that's why it was very important for our project that we conducted a number of stakeholder events to make sure that folks were informed and knew what we were up to and were broadly supportive of the project. So this was a long-term collaboration between the St. Ledger Lab and Abdoulaye Diamante's lab. So what was our experimental setup? This is my colleague Etienne Bilgo. He is looking for mosquitoes on this cow. And so we tried to keep things as simple as we could. We had behind him black cotton cloth that we would soak in oil that had fungal spores. And then we would let that dry overnight, and then we would hang it up in a wall inside of those houses that I showed you. That was our only way of introducing this fungus, was just as a treatment hung on the wall. And so for this first experiment that I'm going to show you, we would come in, we would release 100 mosquitoes into the compartment. They would be able to fly around wherever they wanted to go. Female mosquitoes, spoiler, they want to feed on blood, so they would go to this cow. And then the mosquitoes, after they blood feed, they want to rest somewhere where they're protected. And so usually they go somewhere shaded, but instead they go to this black cloth because we chose the black cloth. And so by releasing these 100 mosquitoes, they could go anywhere they wanted in the compartment. And then we would come back the next day and collect all of the mosquitoes that we released by hand. So there were lots of teams involved in following up and doing this work that I'm showing you. And I want to acknowledge there was a lot of hard work to do that. Imagine trying to pick a mosquito off this cow. And so in our compartments, we either had an oily sheet, a sheet with the wild type fungus, or a sheet with the transgenic fungus. And this is what we found. And this is that study that Hannah mentioned, the transgenic metarhizium rapidly kills mosquitoes in Burkina Faso. And in blue, in all of my figures, is the control. In red is a RFP marked wild type. We wanted to be able to track our wild type, so we gave it a fluorescent marker. And in green, we have the hybrid toxin expressing fungus. This is a fungus that once it makes it inside of an insect, it turns on that blue mountain funnel web spider toxin. So what we found, just like we saw in the laboratory, is that mosquitoes are killed faster, and more mosquitoes are killed by the transgenic fungus just by hanging that cloth up there. And so you can see this figure levels out around like 25%. That is our sort of understanding of that 25% that didn't get infected. They just didn't interact with the cloth. And that happens in the field. So having 75% of mosquitoes that were released and could go anywhere they want, killed by that fungus, was a really exciting, promising result and recapitulated what we were seeing in the lab in semi-field conditions. But we built this giant facility. We're not just going to conduct one experiment and be like, you know. So we conducted a number of follow-up experiments trying to understand the biology of the mosquitoes and how they're affected by this toxin. And so I'm just highlighting a photo here. Up here in the top left corner, this is an insect which is experiencing a phenotype called knockdown. Typically, when mosquitoes are exposed to a chemical insecticide, they will fall from that chemical insecticide because it's very toxic to them. But mosquitoes that are resistant won't have that phenotype. What we found is that when mosquitoes are infected with metarhizium, the wild type and the transgenic ones, as they get sicker and sicker, that phenotype comes back. And the ability of chemical insecticides to kill those mosquitoes is restored because the mosquito is sick from the fungus. For all of these, we found that the transgenic fungus is having a much larger impact. The wild type fungus is not doing too bad either. We also were able to establish populations in the sphere. We would release 1,000 males and 500 females in the compartment and allow those to mate and lay eggs and continue to develop in the sphere. What we found is when we introduced our claws into those environments where these mosquito populations are growing, after just 2 generations, the hybrid compartment had only 10 larvae in it, whereas the control compartments were continuing to grow. And so this is starting to get at how it's not just the killing of the mosquito, but it's also these broader knock-on effects on the development of the next generations and their fitness which we're seeing in this system, which is important to characterize, and you kind of need these larger facilities to look at. The other thing, which is massively important, is mosquitoes lose interest in blood feeding. So even before that mosquito hits the ground, it is not going to bite a person or its host. And to transmit disease, mosquitoes have to bite. And so even before we killed that mosquito, we had stopped malaria transmission. In the case of the wild type fungus, or in the case of the transgenic fungus, in just 3 days, we saw a reduction in blood feeding such that they can't fly to and feed on an insect host. These are really exciting findings for our one toxin, but we actually were able to look at a variety of toxins. And if you think of the diversity of spiders, the number of toxins within them, this is a strategy which can be scaled across a number of systems and hosts. The last thing, which we were able to publish just recently, is looking at sexual dissemination of transgenic fungi in the mosquito sphere. So what we did is we released 500 infected males. These are males that were treated with fungal spores. We released 250 uninfected females. We allowed them to fly around the environment. We typically would release these just before dusk, which is right before they like to mate. And so what we would often see is you release them, and then the male mosquitoes would start to form what we call lek. This is sort of a cloud of male mosquitoes that are all flying together and want to mate with a female mosquito. So they're saying, oh, it's time, I'm here, like this is the spot that we have chosen to mate. How they choose that spot is a really interesting thing. But in our case, they would typically go into the corners of our compartment, and then female mosquitoes that wanted to mate would fly through that cloud. And if they decide to mate, they fall out as a couple. And so what this allows us to do in the compartment is actually count the number of couples that we saw, number of mating events that had occurred. And so we did this, we released them, and we saw about 25% of the females mated. And then we would individually recapture them in the morning. This is what we like to do. We like to individually recapture mosquitoes. And then we would look at the mosquito survival over 2 weeks and see, was the male able to transfer that fungus to a female and kill her? And so these are the results we saw. Male mosquitoes, it turns out, if you treat them directly with a high dose of spores, they're going to die. We still saw that the transgenic fungus was killing them a little bit faster, even at these very, very high doses. But in the case of the female mosquitoes, we saw a really interesting pattern, which is that the transgenic fungus, hybrid toxin, killed about 26%. That is roughly the number of couples that we estimated. Whereas the wild type fungus only killed about 16%. So what this is telling us is that if a male that's treated with the hybrid fungus mates with you, probably 100% of the time you're going to die. If it was treated with a wild type fungus, it's a coin flip. Maybe you'll die, maybe you won't. But this showed this idea of using the male, its mating behavior to go and deliver that spore to the female could be really effective and could be a way to combine this with another technology. We were actually able to quantify the number of spores that were transferred and show that they're transferring the same type for both our wild type and our transgenic as well. So this is not a difference in spore dose, it's a difference in that hybrid killing with that toxin versus the RFP not being able to kill the toxin. When we look at the timing of this, what we found is that we can see similar effects over 1 hour, 6 hours, 12 hours, and 24 hours after treatment. We wanted to look into this because if you want to try to incorporate fungi into a technology where you release males, you're probably going to need to be able to move those mosquitoes some distance. And so we wanted to know how long before releasing them can you treat with the fungus and still have that impact. And so you could treat a male mosquito and then hang on to it for a day and still release it and have that effect that we saw. This was a little surprising to us, but also very promising and fits with the timing of how long it actually takes for the fungus to germinate and move into the insect. And so by day 2, where we start to see it drop off, the fungus has probably already burrowed into that male insect and is not going to be transferred. The last thing that we were able to look at with this collaboration is actually how symbiote status impacts infection. So we're starting to look at how Wolbachia, which is a common endosymbiotic bacteria that many mosquitoes harbor, impacts mosquito infection. And what we found is that, I'm not going to walk you through every figure here. What we found is that the different types of Wolbachia may protect different amounts. But when we increase the temperature, the Wolbachia strains have an increased protection. And so the temperature and the symbiotic status work together to determine if that fungus is going to be able to infect that insect. Very interesting sort of broader combination. The microbiology and thinking of these mosquitoes as sort of a whole biont and how that impacts the outcomes that we see. Part of the reason we wanted to test 30 degrees is because while 26 degrees is a temperature that we might use in the laboratory or rear mosquitoes at, 30 degrees is much more similar to field conditions. And so we're seeing Wolbachia protects mosquitoes from fungal infection at field temperatures, which is concerning and also very interesting. I talked about this one project, but I want to highlight that these fungi can provide a suite of options for biotechnology. We can use them to target a number of different vectors. They can express RNAi, and we can use the ability to routinely modify them to disrupt host symbioses and create things that are limited essentially by your imagination. And so, like I mentioned, you could scale it across a number of different toxins. You could swap out which fungus you're using. But when I think about this technology, I think about that broader sort of system of plugging these different aspects of DNA and toxins together. But you can move it, and the potential of it goes far beyond just this stream. I want to close talking about some particular fungal projects. And so this is a fairly typical approach for entomopathogenic fungi. Can I get rid of this? Yeah. This is sort of a broader summary slide of a pathogen that we isolated in the Casson lab for elongate hemlock scale. Elongate hemlock scale likes to live under fir needles. It's a problem for Christmas tree growers. And so we proposed just looking for a fungus that infects them as a biocontrol option. And then essentially immediately when we got there, Matt and I, in that picture there, found these little orange cushions underneath needles. And it turns out this was a fungus Canodia crella, Canodia rostrata. And so in our lab, we were able to sequence it. We were able to publish on its contacts with other fungi, mycologia, and I think it is a promising biocontrol option for elongate hemlock scale. And this sort of approach to identifying fungi, where the scientist goes out to the area, they have specialized knowledge, they find it, they come back, and we try to apply that for biocontrol, is fairly typical. But another approach that I've been using is community scientist surveys. I'm fairly active on iNaturalist, particularly with these entomologic Israelis that I have been talking about. People on iNaturalist know that I'm obsessed with them and I work with them. And so I'll get tagged in findings that people have. So this is an example of a comment where someone said, I don't know what this is, but Brian might be interested, especially if you can send a specimen. And so this scientist in Ohio, with no idea what she found, reached out to me and found what I think is a new species of entomophaga. This is infecting these little leafhoppers. Here you can see these really fascinating ballistospores that are produced by Entomophaga israelis, where these little spores on the edge are released from the cadaver as sort of a cannon. And so all of these intersegmental membranes, full of these fungal cannon shooting spores and infections. And so I received this in the Casson lab. I started to work with it. I was trying to isolate this entomophaga, but they're very hard to isolate. And so I sent some off for that cross-sectioning that I showed you earlier. I sent some for broader sequencing so that we can look at them. But you can't plan on being able to isolate them. It's not a guarantee. And so I tried sort of threw the kitchen sink at it, literally. But when I got these images back, there was something really bizarre. Maybe some of you have thought this. There's a bunch of scientists in this room. But there are actually a second cell type interspersed inside this insect host. And so aside from these massive circular entomophaga, what we call hypholungs, there are these little yeast-like cells that are interspersed among those pathogenic particles. When we do differential stains, you can see they really stick out compared to the other ones. And so it was clear to us that there was a second fungus that seemed to be a major member of this community. And so I tried to isolate that. And we were able to grow what we now know is a yeast-like symbiote. This is something that is a symbiotic friend of that leafhopper that is helping it to have essential vitamins and essential amino acids. And this is a story that we have seen in other insects as well. But culturing them and working with them is not very common. And so when I was able to culture this, immediately I was like, okay, I want to sequence the genome. I want to understand what we can about this organism, which I may only have for a short amount of time. But you can see for a fungus, particularly what should be a filamentous fungus, it's growing in a really bizarre way. It kind of has these weird sinuses, sort of gooey looking. Fungi typically look fairly dry. And so this is something that even just by looking at, you can see this has changed something weird is going on. And so when we sequenced this genome and compared it to the other YLS that are available, just a handful, what we found is this story of a reduction in genes in that fungus. Through this domestication, through that fungus being recruited into this leafhopper as a symbiote, it had lost genes and also its genome has gotten smaller. This is really bizarre for me because I'm used to working on massive fungi, and so having tiny little fungus to work on was a fun change. And when we looked at all of these different fungi that are yeast-like symbionts that have these sort of mutualistic interactions with insects, what we found is that while they all show this reduction, this loss, the only thing that they have in common, one gene group, is this glutamine-dependent NAD+ synthetase. This is highlighted in orange here, and then you can see the yeast-like symbiote that I was working on, which is called BLYLS, and all these YLSs have this in common, but they have this one gene group. And so what this is telling us is that glutamine is likely mediating this interaction. It seems to be an important thing that they have to maintain, even though they're losing lots of other genes. So it's a really interesting, complex story. It seems to be a process that's happening in lots of insects. So I was really lucky to dig into just, like I mentioned, there's lots of other insects. We've seen this. And cicadas are a classic example, where in Japan, they sampled 24 different cicada species. And 15 of those 24 species had these yeast-like symbionts instead of the canonical bacterial symbiont that most folks assume cicadas have. And so what we see is that when cicadas have this yeast-like symbiont inside of them, they have evicted one bacterial symbiont and are taking the place of that bacterial symbiont. So here we sort of have a turnstile of microbial partners where these fungi can stand in and play out. We're just starting to understand and characterize this system. We see very similar stuff, microbial communities and a wide range of insects. And so I have started to think, in this case, it's Asian citrus psyllid on the left and one of the cicadas from the Japanese study on the right. I have really begun to think of these specialized symbiotic organs as a microbial battleground, where microbes are constantly trying to vie for what is probably very attractive and calm real estate inside of an insect. Naturally, insects will pass this bacterium onto the next generation. So if you're able to set up a home here, then you're going to find yourself in lots and lots of insects down the line. And so the way that I frame this is as organisms existing on this frame of mutualist to pathogen, this sort of continuum, how we think of symbiosis, but also adding this temporal context, where as our insect pathogen fungi move through particular stages, how mutualistic or pathogenic they're behaving shifts over time. And so in the case of these yeast-like symbiotes, the most simple explanation would be that the yeast-like proliferation that we naturally see in entomopathogenic fungi, they're being locked into that phase. And when they're locked into that phase where they only grow as yeast, then they can colonize these tissues without being pathogenic or causing major problems. This is a broader sort of theory of how that can come together. There's so much we have to take apart in this. Okay, so when I was in the Castle lab, I was able to dig much more broadly into fungi, and I did promise you fungi as a particular topic, and so I'm going to dig into two different feeders of fungi. The first are fungus-feeding millipedes. Somebody heard of fungus-feeding millipedes? Well, there are millipedes which are highly understudied, which have formed symbiotic relationships with fungi. They will exclusively feed on fungi. They have piercing mouth parts to actually suck the juices out of fungi. It's a really interesting long-term evolutionary partnership. And there are basically a couple papers describing that true. And so in the Casson lab, here's Matt Casson and his student Angie Macias. Angie spearheaded efforts to try to characterize this broader community and understand them more. And so here they're showing some Brachycytidae lecontii. This is called the pink feather millipede. And they were initially just trying to ask, okay, well, what fungi are there? How can we understand them? So I supported Angie's project here, and I just want to share some high-level findings for you. First is, these fungus-feeding millipedes are fairly divergent. It's a very old relationship, and it's something that they have been maintaining for a very long time. And so we expect that as we dig into these symbioses, we're going to find some really interesting coevolutionary patterns or relationships between these fungi. But even which fungi they're feeding on was unclear in the literature. And when Angie had started these studies, there was one study that said they might feed on this fungus. And that was all we knew about what they fed on. And so this relationship, particularly with these very early terrestrial animals, is something that we wanted to understand more deeply. So when we look at Brachycytidae lecontia, look at this beautiful organism. Those little white dots to the left and right of it are actually the juveniles. They don't turn pink until later. And when we sequence these, the ITS sequence to look at which fungi are present across all of these different sites, we were able to characterize using the AMTK pipeline this broader sort of pattern of the microbiome. This work is not drawing particular conclusions or going in a direction. We are just establishing baseline. What do we expect to see? What are we seeing on these millipedes? And so this work is working its way through publication right now. But we're able to say, like, oh, we've seen this on this host before. This was present across many different sites. That might be one which is actually related to them. One of the things that we saw in here, which is something that we would expect, is that there were a number of polypore fungi. We'll often see them feeding on these crust fungi when we collect them. And so finding them on Flebia, for example, the one that I highlighted there, fits with our observations and expectations and just shows that this molecular work is connected directly to the natural history work we're doing as well. And so these broader patterns sort of painting that picture for future experimental work and picking apart these interactions. But that's just one species. There's so many of these millipedes across the US. And so in the Casson lab, they were able to secure a Nat Geo Explorer grant where we were able to go to all of these different sites across the US and collect dozens of different millipedes. And so we collected millipedes to understand the chemical profile, we collected millipedes for that sequencing, and we also collected millipedes so we can culture fungi directly from them. And so these studies are going to open up the door for a broader understanding of the Colocognatha. And I wrote for the Entomological Society of America, a sort of like ode to flipping logs and finding millipedes underneath it. And so if you want to see how nerdy I am, you can read that article. Final thing that I want to share with you are another arthropod which has decided only to feed on fungi, which are ambrosia beetles. These are organisms that I got to work with my postdoc, Elizabeth Moore, and we were working in the apple system. So these beetles in New York are a problem for apple growers because when apples are flooded, then these beetles will spread into the apple system and potentially kill limbs or the tree. And so understanding their biology and understanding ways that we can control them with fungi was a key focus of Elizabeth's work in my lab. And we worked on a range of different ambrosia beetle species in the apple system to try to understand which fungi can we bring to the table against these beetles. Now, the mutualistic side of ambrosia beetles are their fungi, the ambrosiella. And these fungi produce these sort of chain-like structures that are conidia, spores, that the beetles can feed on. And when they feed on these spores, fungus will produce more conidia, and it will be this sort of like constant food source for the beetle growing up. It's a really interesting system, and even the name of the fungus, ambrosia, refers to the food of the gods, sort of beautiful white structure that they get to live on. And they feed on all of their different developmental stages, all the way through larvae to adults, only on this fungus. And so it makes them very hard to control because they're living and feeding inside of wood. So you can't just spray an insecticide inside of the wood. We have to come up with strategies where we can kill them the brief moments when they're out of wood, spreading into new trees. When trees are stressed by water, they will respond by upregulating ethanol. And that ethanol in the tissues is something that the beetles are attracted to, and is something that those fungi have been shown to enjoy. And so this highly ethanol environment is key to rearing and broadly understanding working with these fungi. And so this sort of context of flood stress trees is how we approach this work, again, on an SCRI, trying to support New York apples. And so Elizabeth loved to work on methods, and we developed a number of ways of working with these tiny beetles and culturing them, and also culturing the fungal symbionts. All of those individually are difficult things, but we needed to do that to be able to ask some of these experimental questions. So she conducted plate assays where we treated beetles directly with fungi. The technique that we developed, I love this as a technique. We would pour the beetles into a falcon tube, and then we would pour fungal spores in the falcon tube, and we would vortex. And they become very infected. And so we can do this to just like broadly screen, like, is this going to kill them? But then we were able to move into what we call bolt assays, where we actually have large sections of wood, and we allow them to more naturally develop inside of a bolt. And for these, we can either treat the bolt directly, or we can treat the beetles. We were also able to use these sort of direct treatments of beetles to look at how that ethanol concentration is affecting our system. So we could treat the wood in our experimental system with different amounts of ethanol to understand how that affects broader development. And then we also couldn't help ourselves. We did a little just direct mycology to understand the biology of the fungi. And so fungus that I'm going to share with you right now or focus in on Bavaria bassiana. This is a widely used, massively important commercial product. And we're using Botanigar in these studies. So here, you look really hard, there's a beetle under all those fungal spores. It's been killed by Bavaria. Bavaria grew out, sporulated on the outside. Okay, so, wait, there's one more. Okay. The environments that I want to walk you through, I want you to sort of keep in your mind, are we have Petri dish assays where there's no ethanol. And we have bolt assays where we can soak the bolt in ethanol. And then we have some assays where we treated the beetle directly. And we have some assays where they're picking spores up off the bolts. We treated the bolts, and as they sort of walk around it, where they burrow in, they can pick up spores there. And in particular, we're using Bavaria bassiana, which is a particular strain that we isolated in the lab from that commercial product. And so what we found when we looked at this fungus is that when you treat them directly and you put them in a low ethanol environment, those beetles die. They're susceptible to the fungus is able to kill them. However, when we treat the beetle and put them in a bolt, only half as many die. And so just by naturally engaging in their environment, half of them are protected from fungus. If we allow them to pick the fungus up off of the wood, almost none of them die. And so clearly this ethanol environment is getting in the way of the fungal infection that we would naturally expect from those profile assays. And so we decided to look at ethanol. showed you those ethanol, those different concentrations of ethanol. And we got a new bag of our biopesticide, and we found totally different results. We found that even at high concentrations of ethanol, they were killing the beetles. And so this led us to this broader understanding that it's not, it is the identity of the fungus, it's the strain, which is leading to the sort of difference in results that you're seeing. And so this fungus, EM004, the 044, seems to be able to handle that ethanol environment, even at these higher concentrations, and still kill those beetles. And so to sort of test this idea that maybe this fungus can handle ethanol better, we conducted some studies looking at these two fungi. So over here on the left, we've got our ethanol intolerant one, our ethanol tolerant one on the right. This is how they grow with no ethanol. This one just grows less. It's not doing very well. It grows smaller than that one. But you can see when we add ethanol, there's very little effect on our ethanol tolerant one, but our ethanol intolerant one massively decreases not only its growth, but the number of spores they produce as well. So this is something which we think sort of supports that broader idea, but also highlights that both of these fungi, which are behaving very differently under these conditions, were isolated from the same commercial product. And so strain matters, and the abiotic preferences, tolerance of these fungi is really important to bake into how we understand and are testing and optimizing these commercial applications. And in the case of ambrosia beetles, because of that high ethanol environment, it's really important to consider this context that allows us to notice these fungi. And so with that, say thank you so much for listening to me, talking about all of these different insects and fungi, and I would be glad to take any questions.